Okay, so welcome everybody. I think it's noon here in New Haven. So it's my pleasure to welcome all of you again to a new Global Immunotalk. Just briefly, I would like to remind you that we have developed this series with the goal of benefiting and inspiring immunologists across the world in an egalitarian manner. And the great thing is you can view this from wherever you are, just through this Zoom or in the YouTube channel. And so this really increases the opportunities for all of us to learn about fantastic science. So we, of course, cannot do this without our extraordinary speakers. We wanted to expand our speakers in order to have representation of also early career scientists. And today we're delighted to welcome Juan Cubillos-Ruiz, who is going to be introduced by Elena. But let me remind you that next week we will have also Dan Littman, so stay tuned for Dan's talk next week. With this, Elena, now it will be great to have you introduce Juan.
Yes, thank you so much, Carly. So yeah, I feel really excited with this initiative to incorporate young emerging leaders into our Global Immunotalks program. And I cannot think of a better way to start with this than introducing Dr. Juan Cubillos-Ruiz. So Juan is another international global immuno speaker, in this case, original from Latin America, as Juan was born in Bogota, Colombia. He received his bachelor degree in microbiology from University of Los Andes in Bogota, Colombia in 2004. And in 2005, he moved to the US, where he earned his PhD in immunology at Dartmouth Medical School in 2010. Juan then pursued his postdoctoral training at Harvard University and Weill Cornell Medicine in the laboratory of Dr. Lori Glimcher, where his research focused on understanding how endoplasmic reticulum, ER stress response pathways, influence the immunobiology of cancer. In 2015, Juan was appointed as an assistant professor. And since 2018, he has been William Ledger, MD, Distinguished Assistant Professor for Infection and Immunology in Obstetrics and Gynecology and member of the Mayo Cancer Center, all at Weill Cornell Medicine. Juan is an unquestionable emerging leader in the field of tumor immunology, who has pioneered the study of ER stress responses as key regulators of cancer immunity, anti-cancer immunity. This work demonstrated that adverse conditions in the tumor microenvironment provoke severe ER stress in leukocytes as a mechanism that promotes immune evasion and cancer progression. His laboratory combines immunology, molecular biology, genomics, and nanotechnology to identify, understand, and disable the mechanisms that tumors use to inhibit protective immune responses. So I cannot wait to learn more about all of this in Juan's talk today. He has published his work in top-tier journals, including Cell, Nature, and Science, among others, and has been very successful at competing for extramural funds to support his research program. And he has already been honored with many important awards. For example, in 2015, Juan was elected to the Ovarian Cancer Academy of the Department of Defense, and more recently received the Pershing Square Sean Prize for Young Investigators in Cancer Research, among other honors that are listed in his CV. On a personal note, I met Juan when he visited UCSD about 3 years ago, and I was not only captivated by his amazing science, but also by a story he told us on his experience as being the only Hispanic graduate student in his class at Dartmouth, and how this fueled his desire to contribute to diversity. So I think Juan is not only a terrific emerging leader in tumor immunology, but also a fantastic role model for underrepresented minority students and young scientists. So Juan, thank you so much for being here. It's a pleasure to have you here. And thank you for your efforts to prepare this talk.
Well, thank you, Elena and Carla, for the invitation. I think it's a real privilege and honor to be presenting, at the Global Immunotalks. So it's a wonderful opportunity for me to share our research with the community in this manner. Thank you for the introduction as well. I really appreciate it. And I'm excited to deliver the talk. So fantastic.
Yeah, Juan, before we let you deliver the talk though, you are our youngest global immuno speaker so far. So we think that you have more fresh the memories on how it is to start your independent research program, and wonder whether you could share your advice for young student postdocs that are considering establish their independent laboratory.
Sure, absolutely. So very briefly, just think that it's going to be completely different. It's going to be a new world. It's something that no one has trained you for, neither in grad school nor during your postdoc. But it's extremely exciting. It's challenging, and the only advice that I would give PhD students and postdocs thinking to continue in academia is exactly what my former mentors told me: is to think outside the box. Don't be afraid of taking risks. I think that's the fun part about doing research in academia, is that you can explore your creativity. Of course, you can have a balance between safe projects in the lab, solid stories that will lead to paper 1, 2, and 3. But I think the cool part of what we do relies on taking risks, and postulating these high risk, high reward questions, that maybe can change the way people think and can make a huge impact in the field. So that's the only advice.
Fantastic. Thank you so much, Juan, for sharing that. And if you'd like to share your screen.
Perfect. I'll do it now. Great. Okay.
Perfect. We can
Perfect.
see it now.
Can you see it now?
Great.
Okay, wonderful. So thank you again, Elena and Carla, for the invitation. I'm thrilled to be here. I just wanted to share a little bit of our research during the last 5 years, trying to understand how intratumoral immune cells sense and respond to different stressful conditions in the tumor microenvironment, and how we can exploit this knowledge to hopefully create new forms of immunotherapy. So as we all know here, immune cells have a remarkable capacity to recognize and eliminate cancer cells, and this has inspired many therapeutic approaches trying to harness this activity, and has now led to the development of multiple forms of immunotherapy that are making a huge difference in the way we treat cancer. However, cancer cells, and especially tumors, have developed different strategies to counteract the function of immune cells. And actually, there's a huge unmet need to understand the mechanisms by which tumors can inhibit or block the function of immune cells. So one of the key mechanisms by which tumors block immune cell functions is actually by creating harsh microenvironments where immune cells cannot operate. So it is well known that the tumor microenvironment is characterized by multiple stressful conditions and factors, ranging from nutrient deprivation, hypoxia, low pH, accumulation of lipid mediators. There's also accumulation of reactive oxygen species, and there's an ongoing metabolic competition between the cancer cell and the immune cell, and this has been described by elegant work from multiple labs around the world. What we found, and others in this field, is that all these different conditions in the tumor microenvironment actually cooperate or converge to compromise a critical function in the cell, which is actually protein folding in the endoplasmic reticulum. So if you remember, the endoplasmic reticulum is a central organelle where all the transmembrane and secreted proteins will be folded, modified, and exported. So this process is tightly regulated. What we have found is that all these different conditions in the tumor can create a state of endoplasmic reticulum stress characterized by the accumulation of misfolded proteins in this compartment, which we believe is a central mediator of immune cell dysfunction in the tumor. So that's exactly what we're going to be talking about today during our Global Immunotalk. So basically, the ER is in charge of lipid biosynthesis, calcium storage, but as I mentioned before, most importantly, protein folding, disulfide bond formation, and end-link glycosylation, which are essential modifications needed for proteins to be properly folded and acquire their right functional three-dimensional structure. So what we and others have found is that the different factors that I mentioned in the tumor microenvironment can greatly compromise the protein folding capacity of the ER, and therefore block the normal processes that occur in this organelle and drive a cellular stress, state known as endoplasmic reticulum stress that is characterized by the accumulation of misfolded or malfolded proteins. So when this happens, the cell actually can respond to the stress. And cells have evolved a very interesting and complex adaptive signaling pathway called the unfolded protein response, also known the ER stress response, which is controlled by 3 ER resident proteins that operate as sensors of ER stress. So when these proteins are activated upon accumulation of misfolded proteins, they trigger secondary adaptive responses like the unfolded protein response that tries to reinstate the protein folding capacity of the ER. It also induces cellular reprogramming, mostly mediated by transcriptional reprogramming, but at the same time, this pathway, the ER stress response, can trigger autophagy and apoptosis. And interestingly, what we know so far is that in the tumor microenvironment, the magnitude and the level of ER stress responses can greatly determine cell survival and cell fate and cell functions in the tumor. So for example, what has been demonstrated is that a low or moderate level of ER stress that is persistent and that is usually driven by oncogenic signaling or metabolic alterations on the microenvironmental conditions that I mentioned can actually promote tumor growth. This is what we call the protumorigenic ER stress responses that facilitate proliferation of cancer cells, angiogenesis, chemoresistance, metastasis of primary tumors to distal sites, and additionally immunoregulation. But at the same time, when the ER stress is extremely high and when there's unresolved ER stress, especially upon treatment with some forms of chemotherapy, this ER stress can actually be exploited to kill cancer cells. And that's what we call a lethal unresolved ER stress that induces apoptosis, and in some cases is useful to trigger immunogenic cell death and anti-tumor immune responses. So my lab focuses on the most evolutionarily conserved arm of the ER stress response, which is the IRE1 XBP1 branch. So let me tell you a little bit about the molecular biology and the mechanisms of action of this ER stress sensor. So IRE1 is an ER resident protein that sits on the membrane of the ER, and it has a luminal domain and also a kinase domain and an RNase domain that are facing the cytosolic region. Interestingly, under normal conditions, when there's no accumulation of misfolded proteins, IRE1 is usually not activated. But when there's accumulation of misfolded proteins, this sensor dimerizes and undergoes autophosphorylation via its own kinase domain. And the kinase domain, this event actually induces activation of the endoribonuclease domain of IRE1. So it's a beautiful process by which the detection of misfolded proteins triggers these 2 independent domains. What is important about this process is that the RNase domain of IRE1 cleaves a 26 nucleotide region in the unspliced XBP1 mRNA that is in the cytosol. So this is actually considered an unconventional splicing event. And then upon excision of these 26 nucleotides, XBP1 the spliced form is religated, and now it's able to code for the functionally active transcription factor XBP1. In the absence of IRE1, and especially its RNase domain, XBP1 cannot code for the functional transcription factor. And you can actually detect this very easy in the lab just by doing the splicing assay, where you can see that upon stimulation with ER stressors, you see the emergence of the second band of the spliced isoform of XBP1 that codes for the transcription factor. So XBP1 is a multitasking transcription factor that has UPR canonical roles. For example, is a driver of the expression of multiple chaperones, foldases, ERAT components, and multiple proteins that participate in restoring the quality control and the protein folding capacity of the ER. But what we and others have found over the last decade is that XBP1 as a multi-transcript transcription factor can actually drive or control other UPR-independent processes like inflammation, lipid metabolism, and mitochondrial function. So this is really exciting, you know, to realize that XBP1 has UPR canonical functions and also UPR independent functions that can influence the function of immune cells in health and disease. And so for the last 15 years, it's been well established that tumors, especially cancer cells, can exploit the sustained ER stress responses to promote malignant progression and tumor growth. So in the case of IRE1 alpha and XBP1, it's been well established that this overactivation of the pathway can drive chemoresistance, EMT, metastases, angiogenesis, and tolerance to hypoxia. So therefore, it's an adaptive pro-survival mechanism that allows cancer cells to thrive and to actually promote tumor growth. And this has been seen in multiple tumor types, ranging from breast cancer to multiple myeloma and pancreatic cancer. Okay, so the question we had when we started this research a long time ago was, as immunologists, we wanted to understand if maybe the immune cells that inhabit the same tumor microenvironment could be experiencing ER stress. And we really wanted to understand whether the ER stress, if so, that the immune cells were experiencing was pro-tumorigenic or actually required for mounting anti-tumor immune responses. So this is a work that I started as a postdoc a long time ago in the lab of Laurie Glimcher, and basically we focused on ovarian cancer as a model system, because ovarian tumors are extremely aggressive, they're highly immunosuppressive, they're full of dysfunctional immune cells that cannot mount protective responses against these malignant masses, and also because it's extremely aggressive. I'm sorry to show this graph, but I just wanted to show you how terrible this disease is in terms of its metastatic capacity and how resistant to standard treatments and refractory to immunotherapy is. So we had previously characterized that dendritic cells are dysfunctional in this model, and we wanted to understand if dendritic cells, which are central mediators of anti-tumor immunity, were experiencing ER stress at tumor sites in the tumor microenvironment. So basically what we did was to actually isolate these dendritic cells from different human ovarian cancer specimens and also from mouse models of metastatic disease, and then determine if maybe that pro-tumorigenic or dysfunction that these dendritic cells experience or that show in the tumor was controlled by potentially ongoing ER stress responses. So surprisingly, when we isolate these tumor-associated dendritic cells, we found severe overactivation of the IRE1 XBP1 pathway. So here I'm just showing some representative data from different ovarian cancer tumor models where we isolate tumor-associated dendritic cells, or also splenic dendritic cells, or also from metastatic models based on the IDH system. You can see that in all cases, these dendritic cells show significant upregulation or abundance of the spliced functional isoform of XBP1, while normal dendritic cells in the spleen or lymph node or bone marrow of mice that didn't have tumors actually showed significantly lower levels of XBP1 splicing. And this is quantified here by qPCR, where we can selectively determine the expression of the XBP1 S isoform. What was most striking was actually the results that we got by Western blot when we were interrogating the expression level of the actual nuclear transcription factor using nuclear lysate. So this is XBP1S in tumor-associated dendritic cells was significantly upregulated and actually stabilized in these dendritic cells, indicating that indeed these cells, just like cancer cells, were also undergoing constitutive or sustained ER stress responses. So then the next question was to try to understand what is the role of XBP1 overactivation in these dendritic cells. So we had access to elegant conditional knockout mouse models where we could selectively delete the pathway in specific cell types. Back in the day, to determine the role of this pathway in dendritic cells, we used the CD11C3. And then what we did was to actually exploit this system to develop ovarian tumors and determine the progression of these malignant masses in mice that didn't have XBP1, specifically in CD11C positive cells. So this is the first genetic evidence that we got back in the day showing that XBP1 expression in an immune cell, not in a cancer cell, which was shown before, is actually critically required for optimal tumor progression. So here we're showing the induction of ovarian tumors in gems of ovarian cancer in XBP1 fluxed control mice or XBP1 fluxed CD11C3 mice. And you can see here there's a significant decrease in tumor growth and also in tumor onset. Okay, so we confirmed this in multiple models, also in metastatic models of disease. But interestingly, the same effects or similar effects were observed when we deleted the upstream activator of XBP1 IRE1 alpha, demonstrating that it was the canonical IRE1 XBP1 linear pathway that was implicated in controlling this protumorigenic process in dendritic cells. So what was interesting was to actually then when we immunophenotype these animals and when we did the same experiments in other models of disease. So this is a model of metastatic ovarian cancer where you can inject ID8 cells directly through the peritoneal cavity and then generate these peritoneal tumors that recapitulate human disease. So in this case, in this context, ablation of XBP1 also reduced tumor growth and metastasis. So this is a typical peritoneal carcinomatosis that we observed in wild-type animals, but there was a significant reduction in the number of peritoneal lesions and peritoneal tumors in the animals lacking XBP1 in dendritic cells. They also accumulated ascites more slowly, demonstrated reduced tumor-induced splenomegaly, and survived longer than the controlled gene XBP1 sufficient counterparts. So we were excited about these results, and we decided to try to understand the mechanisms by which these dendritic cells were inducing potentially a delay in ovarian cancer. So the exciting part of the story comes when we try to analyze the function and the phenotype of intratumoral T cells in these mice. So it turns out that this is the normal activation of status of CD4 and CD8 intratumoral T cells in a wild-type animal. So you can see here that only 6 to 5% of these intratumoral T cells can express interferon gamma at tumor sites, but the situation was different when we isolated T cells from tumor-bearing mice that lacked XBP1 in dendritic cells. And actually the proportion of activated effector T cells in the tumor microenvironment increased significantly, and this occurred in the 3 independent models of ovarian cancer that we tested. So I wanted to summarize mechanistically what we found over the last years. Basically, the driver of ER stress in the tumor-associated dendritic cells is the accumulation of reactive oxygen species. This ROS that is found intracellularly promotes lipid peroxidation in the cell, and this lipid peroxidation process leads to the accumulation of byproducts, which are reactive aldehydes, that can diffuse into the ER and modify some of the ER resident proteins, especially chaperones, to cause protein misfolding and accumulation of these unfolded proteins. So this is the trigger of the IRE1 XBP1 pathway. So XBP1 results persistently overactivated. It targets many genes involved in the UPR, but what we found was that, interestingly, XBP1 also induced expression of many lipid biosynthetic genes and genes implicated in lipid droplet formation. So these dendritic cells undergoing a persistent ER stress in the tumor accumulated a lot of lipid droplets intracellularly that by different mechanisms blocked antigen presentation to T cells. And this was actually solved mechanistically by other groups, showing that these lipid droplets can actually sequester some of the chaperones that are required for optimal antigen loading onto MHC class I molecules. So XPP1 deficient dendritic cells in the tumor showed enhanced antigen presenting capacity due to restored lipid metabolism. They were more effective at activating T cells, and they promoted memory anti-tumor T cell responses. But the story didn't finish there. So when I started my lab, I really wanted to further investigate the mechanisms by which XPP1 and IRE1 may be controlling the function of immune cells in the tumors. So this was a project of a super talented grad student in the lab who graduated last year, Sahil Chopra, and he really wanted to determine if maybe this ongoing ER stress response driven by IRE1 XPP1 beyond controlling antigen presentation could be also enhancing the expression of immunosuppressive factors by these dendritic cells and other myeloid cells in the tumor. So basically, again, to summarize, this is what he published last year. He found that in different myeloid immune cells, and especially in tumor-associated dendritic cells, ER stress and the overactivation of IRE1 XPP1 can lead to the expression, or is required for the expression, of two critical enzymes that are required for the biosynthesis of prostaglandins, which are COX-2 and MPGAS1. So basically, XPP1 as a transcription factor was able to bind the promoter regions of COX-2 and MPGAS1 simultaneously, and then significantly enhance the expression of these two enzymes to drive the generation of PGE2. And interestingly, there has been many reports demonstrating that PGE2 is the mother of immunosuppression in cancer. So this is a very interesting review published in 2018 demonstrating the multiple immunoregulatory functions of PGE2 in cancer, ranging from recruitment and differentiation of MDSCs, switch macrophage function towards immunosuppressive types, and also the differentiation and migration of Tregs. So we went back to our models of ovarian cancer. We isolated tumor-associated dendritic cells that were wild type or lacking XPP1, and we found that indeed these dendritic cells had a significant reduction in their capacity to make prostaglandins, and this correlated very well with their incapacity to generate FOXP3 Tregs at tumor sites. So this is a normal Treg generation capacity of wild-type tumor-associated dendritic cells, while the XPP1 deficient dendritic cells, again, had a significant reduction in that capacity. Okay, so we found then that this is conserved in humans as well, so we're very excited about these results. So in here, we're using primary human dendritic cells, and we optimize a very cool CRISPR-Cas system based on the transient electroporation with ribonucleotide protein complexes. And we were able to edit or delete XPP1 or IRE1. And we found that when we delete this factor in ER stress dendritic cells, again, there's a significant reduction in their expression levels of COX-2 and MPGAS1, which then leads to a reduction in the expression of PGE2. So it seems that this is conserved in the human, in the tumor in the mouse tumor microenvironment, and also in human dendritic cells when they're exposed to factors that drive IRE1 XPP1 activation. So this is the story from the myeloid cell compartment, but there was a lot of research going on how ER stress responses control the function of these myeloid cells in the tumor. And we had published on the dendritic cells, but other colleagues also demonstrated that it seems that MDSCs and neutrophils and macrophages are exploiting the ER stress response to drive pro-tumorigenic and immunosuppressive programs in the tumor microenvironment. But another grad student in the lab, Min-Hyun Song, who graduated 2 years ago and now has her own lab in Korea, wanted to really determine whether T cells in the tumor microenvironment were also facing ER stress, and if these T cells were directly affected by these ongoing ER stress responses, just like dendritic cells. So she again went back to our tumor bank. We isolated tumor-infiltrating lymphocytes, T cells from different human specimens, from solid tumors, from the ascites of ovarian cancer patients, and we also had peripheral blood mononuclear cells. She isolated CD4s and CD8 T cells, and then she determined the activation status of IRE1 XPP1 using the techniques that I mentioned before, especially the splicing assays. So we wanted to understand if this pathway was also upregulated in intratumoral T cells, which had not been shown before. So this is representative data of some of the patient samples that Min-Kyung analyzed. And surprisingly, the intratumoral T cells, and especially those CD4 T cells and CD8 T cells present in the malignant ascites in the peritoneal cavity, which is this fluid full of cancer cells and immunosuppressive mediators, had a significant increase in the expression levels of the XBP1S isoforms. And this is quantified here, even compared with that found in solid tumors and in peripheral blood that we used as control. So it seems that yes, T cells in the tumor microenvironment, at least in ovarian cancer, were also experiencing ongoing ER stress responses and activation of IRE1 XPP1. So she confirmed these findings by using qPCRs for other UPR marker genes, ER stress response marker genes like BIP, CHOP, and she found out almost a perfect correlation between the expression of those additional ER stress marker genes and XPP1 in all the specimens found. But most importantly, she found that there was a significant inverse correlation between the expression levels of these ER stress response marker genes, and especially XBP1, and T cell infiltration into the tumor, into the human specimen. And also, the more XBP1 or the more ER stress marker genes these T cells were expressing, the lower their capacity to express interferon gamma transcript was. So that suggested that IRE1 XBP1 signaling may also be regulating the effector functions in T cells in the tumor microenvironment. So that was Min-Kyung's project, and she wanted to understand the mechanisms by which XBP1 could be co-opting T cell function in cancer. So she had this question, and she exploited the ascites system or microenvironment that we had to determine how ovarian cancer could be inducing this pathway and to dissect the mechanisms. So basically, she isolated T cells from cancer-free women, and then she activated them in the presence or absence of ascites supernatants from ovarian cancer patients. So they were completely cell-free. There was just soluble factors in the ascites, and then she interrogated whether IRE1 XBP1 was induced. So the first finding that we had is that these supernatants induced a drastic reduction in the expression levels of GLUT1 on the surface of the T cell upon activation. So remember, GLUT1 is a major glucose transporter. So when these T cells were exposed to the ascites, they could not induce this transporter at the mRNA level and also at the surface, and they were unable to take up glucose. And we found that the same was true in vivo in the patient sample. So the intratumoral T cells, both CD4 and CD8, had a very, very, very low level of expression of GLUT1 on the surface compared with tumor cells or with other leukocytes in the tumor microenvironment, suggesting that that was happening also in vivo. So she continued these experiments and found that indeed these ascites supernatants were able to induce the XBP1S form in the nucleus of the T cells in a dose-dependent manner. And interestingly, using 2-MBDG uptake assays, so this is a fluorescent glucose analog that can be used to determine the capacity of cells to incorporate glucose. She found that the lower capacity of the T cells to incorporate glucose, the higher the stress responses and the relative expression of XBP1 in these T cells exposed to ascites. So this was very interesting, and it was actually a little bit expected because glucose is not only important for glycolysis. I know that we have devoted a lot of effort and attention to understanding how glucose drives glycolysis, of course, but I just wanted to highlight that glucose is also extremely important for protein glycosylation in the endoplasmic reticulum. So about 10% of glucose that is incorporated by the cell feeds the hexosamine biosynthetic pathway, which then generates the glycans, the glycosylation mediators that drive and link glycosylation in the ER, like UDP GLICNAC, and that are absolutely required for optimal protein folding in the ER and for the proper export of these proteins from this compartment. So basically, the take-home message is that if you don't have glucose, of course, you affect glycolysis and mitochondrial function, but at the same time, you're generating severe ER stress responses by blocking and glycosylation in this organelle. And you can actually see that this happens in vitro quite quickly and could be modeled very easily. So here we have just normal T cells that have been activated under glucose replete or glucose deprived conditions. At 3 hours after exposing these T cells to glucose deprivation, you see a significant increase in the XBP1S isoform and continues to go up progressively. And you can also use, for example, 2DG, which is normally used to block glycolysis by interfering with hexokinase function. But the problem is that when you block hexokinase function, you're blocking both glycolysis and at the same time the hexosamine biosynthetic pathway. So as expected, as I mentioned before, there's a significant induction of ER stress responses. You can see here XBP1S goes up super quickly, and then you can actually target these or inhibit these by using inhibitors of the RNase domain of IRE1 like MKC8866. So just please remember that when you deprive cells from glucose, you're not only affecting glycolysis, but triggering severe ER stress responses. So it's important for the interpretation of the results. So the question after knowing these in vitro results is whether IRE1 XBP1 could be controlling anti-tumor T cell function in vivo. So we took a similar approach, but instead of using CD11C CRE mice, we exploited the CD4 CRE strain, which can be used to simultaneously delete this pathway in both CD4s and CD8 T cells when they're double positive in the thymus. So interestingly, XBP1 deficiency did not alter T cell development, T cell reconstitution, proliferation, cell cycle progression, or helper differentiation in normal naive mice. So XBP1 was not required for the normal development and function of T cells in mice that didn't have tumors. However, the situation was completely different when these T cells were exposed or were undergoing a physiological ER stress responses. So this is just an example. Here we isolated XBP1 wild type versus XBP1 knockout intratumoral T cells from mice developing ovarian cancer, and we profiled them using them RNA-seq. And we found that the absence of XBP1 or IRE1 in these T cells actually caused a significant transcriptional reprogramming in these intratumoral T cells. And surprisingly, the absence of XBP1 in these T cells induced many immunoactivating networks and many genes implicated in the T cell effector anti-tumor function, for example, interferon gamma, CCL5, the CD28, CD69, Fas ligand, as shown here. So interestingly, all these T cells, or the consequence of this reprogramming was that the intratumoral T cells acquired a more pronounced or more robust effector profile. And you can actually see here by intracellular cytokine staining, the XBP1 knockout T cells in the tumor microenvironment had an increased capacity to make interferon gamma. But we wanted to confirm if these functional effects were really antigen specific, tumor antigen specific. So we repeated these experiments and we actually used different ovarian cancer cell lines that express either luciferase or that express ovalbumin. We developed these tumors into either wild type or conditional knockout mice. We waited until the animals developed solid tumor masses, and then we isolated the intratumoral T cells from these specimens. And the TILs were pulsed with BMDCs that had been were co-cultured with BMDCs that had been pulsed with OVA, and then we determined the capacity of T cells to recognize the cognate antigen in response or coming from each one of the tumors. So interestingly, only in the absence of XBP1, the tumor-specific T cells for the OVA antigen, for the OVA peptide, were able to induce maximal interferon gamma secretion compared with the wild-type counterparts in the same system as control. The knockout XBP1 deficient dendritic cells did not induce interferon gamma in response to a non-relevant or irrelevant antigen. So it seems that these T cells were acquiring enhanced effector functions in the tumor microenvironment, and they were kind of refractory to the ER stress that was evoked by the tumor microenvironment. So these consequently resulted in a reduced tumor growth, just like in the case of dendritic cells. So we used different models of ovarian cancer, and we found that when deleting XBP1 or when deleting the gene encoding the upstream activator, IRE1, we could also substantially reduce tumor growth and metastasis in the peritoneal cavity and increase survival in different mouse models, suggesting again that this overactivation of IRE1 and XBP1 signaling in the T cell was also contributing to tumor progression. So I just wanted to spend 1 minute to summarize what we believe is the mechanism of action or why IRE1 is operating this way in the tumor microenvironment and especially in T cells. So on the first hand, we have a happy T cell that is undergoing activation under normal conditions where you have sufficient nutrients, in this case glucose. So glucose could be internalized by GLUT1, and there's sufficient glucose to undergo glycolysis and also for effective protein glycosylation in the ER. So this is a competent T cell that can make interferon gamma and that can mount responses against pathogens, for example, or tumors when possible. At the same time, this T cell has sufficient energy and sufficient glucose to drive mitochondrial respiration, and then in case there's a decrease in glycolysis or in the levels of pyruvate, the T cell can also utilize other carbon sources to fuel mitochondrial respiration like glutamine. So again, this is a competent anti-tumor T cell. But in the context of the tumor microenvironment, we saw two scenarios that may take place. The first one is that there could be very limited glucose availability in the extracellular milieu because the cancer cell is actively stealing the glucose from the T cell. At the same time, like in ovarian cancer, it seems that cancer cells can secrete some tumor-derived factors that actively shut down surface expression of glucose transporters. In either case, the consequence is the same, is that you have very little amounts of glucose in the cell to drive glycolysis and especially to glycosylate proteins in the ER. So this actually drives defective glycosylation, protein misfolding, and then persistent activation of IRE1 XBP1, which cannot be resolved because actually one of the main functions of XBP1 in the UPR is to drive the expressions of enzymes that will promote protein glycosylation in the ER. However, if glucose is not present, then the pathway will be persistently activated or abnormally activated. And this has a negative consequence. What we found is that XBP1S then controls the abundance of glutamine transporters on the T cell. So it actually induces the degradation, and therefore the T cell can no longer utilize glutamine that would otherwise support mitochondrial respiration in the when glucose and pyruvate is absent. So therefore, this T cell becomes dysfunctional and becomes unable to kill cancer cells. So this is our mechanistic interpretation, which can be recapitulated in vivo and in vitro as well. So based on these results, I just want to spend the last 5 minutes, on the potential translational application of this research. So what we and other colleagues have identified in ovarian cancer and in other tumor types is that it seems that this chronic activation of ER stress sensors, especially IRE1 alpha, can orchestrate multiple immunosuppressive processes or mechanisms in the tumor microenvironment. So these are some of them. First, dendritic cells, dysfunction, abnormal lipid accumulation, decreased antigen presentation, and prostaglandin secretion. At the same time, in a T cell intrinsic manner, these T cells can lose metabolic fitness and effector capacity by experiencing abnormal ER stress responses. And other groups have demonstrated that NDSCs in cancer can also exploit the IRE1 XPP1 pathway to express other immunosuppressive mediators that in a paracrine manner can suppress T cell function. So the overall consequence is that you have global immunosuppression upon activation of IRE1 XPP1 in the tumor microenvironment. So we had this translational hypothesis. We hypothesized that maybe controlling IRE1 XPP1 activation pharmacologically could be used to control these immunosuppressive processes and induce anti-tumor immunity to potentially enhance the efficacy of cancer immunotherapy. So we've had a very deep and profound interest in finding small molecule inhibitors that can selectively block IRE1 activation as its upstream mediator. So this is a work that we started here at Weill Cornell several years ago, and that has led to the identification of two lead compounds called WCM907 and 909, which are selective inhibitors of the kinase domain of IRE1, not the RNase domain, but the kinase domain, and that they operate by blocking this kinase domain and therefore suppressing the subsequent activation of the RNase domain and then blocking XBP1 expression. So we have characterized these compounds. We have done a lot of toxicology and tolerance studies in animals, both with tumors and without tumors. And I just wanted to share some of the preliminary data we have. With this compound, we actually can attain simultaneous inhibition of XBP1 splicing in multiple compartments of the tumor microenvironment at the same time. So these are ovarian cancer-bearing mice that have been dosed for 10 days with these compounds. Once per day, our compounds are orally available and have a great biodistribution. So here we're showing the expression levels of the XBP1S isoform in malignant spheroids found in the peritoneal cavity, in intratumoral disease, intratumoral T cells, but also in omental metastatic lesions. And as you can see here in red, there's a significant reduction in the levels of XBP1S upon treatment with the inhibitors. And interestingly, treatment with these inhibitors correlates very well with delayed tumor progression and increased survival. So we optimized different therapeutic schemes where we can treat orally these mice with the compounds, and then we find that this has significant effects in tumor progression. So here we're looking at splenomegaly in these mice treated with the compound, ascites accumulation upon treatment, also the proportion of malignant spheroids and omental tumor burden. So these are the metastatic lesions in the omentum assessed by immunohistochemistry. And as expected, treatment with these compounds can induce a significant increase in survival of mice bearing metastatic ovarian cancer. So we're very excited about these results. Of course, we still have to continue the characterization and understanding of these compounds in the ovarian tumor microenvironment, but we're also trying to use them in other cancer types. And we're also very interested in defining the function of IRE1 XPP1 activation in other tumor types. So we have developed some computational pipelines to predict the cancer types that are potentially exploring this pathway as an immunosuppressive mechanism. We've seen that lung cancer, pancreatic cancers, and triple-negative breast cancers demonstrate the high overactivation of IRE1 XPP1, and that the levels of expression of XPP1 or IRE1 XPP1 control gene signatures correlate with worse prognosis. So we're also very excited about creating new forms of immunotherapy or combination immunotherapies with these compounds, because there's recent evidence suggesting that the pre-existing levels of ER stress response gene markers, especially XBP1, CHOP, and BIP, correlate with response to immunotherapy in melanoma patients. So this is a very interesting paper by the group of Zev Ronay, published in Nature Communications last year, showing that patients who responded to anti-CTLA-4 treatment actually did better, and they had a significant decrease in the basal levels of the spliced form of XBP1. And this is just the correlation, sorry, showing that those patients that had lower amounts of XBP1 responded better to treatment with anti-CTLA-4. So we're very excited about this and potentially enhancing cancer immunotherapy by targeting ER stress responses in the tumor microenvironment, which multiple other groups are also pursuing. Okay, so in our lab, just to finish up, these are the current directions. So we really want to understand the global immunometabolic and epigenetic programs that ER stress responses control in the tumor microenvironment. We are also exploring how the other arms of the UPR influence the function of adaptive immune cells in cancer. So we have great collaborators, especially at Moffitt Cancer Center. Paulo Rodriguez working on the PERK pathway and trying to understand how the two branches of the ER stress response, IRE1 and PERK, may be cooperating and trying to also uncouple their distinct functions. As I mentioned before, we want to develop new combination treatments targeting ER stress sensors to enhance the efficacy of cancer immunotherapy, not only in the context of immune checkpoint blockade, but we're also trying to generate a next generation of CAR T cells lacking ER stress sensors that we're expecting them to function better in the aggressive tumor microenvironment. So thank you very much for your attention. This is my lab right now. We have a really diverse team of people from different backgrounds, from different scientific training schools. You know, we have fantastic collaborators, and I'm again super grateful to all the different funding sources that have supported our research program. And again, extremely grateful to Elena and Carla for their invitation to present today. Thank you so much.
Thank you so much, Juan. Thank you for sharing your very elegant and impactful work. That was a real treat.
Beautiful, beautiful talk. Thank you so much, Juan.
Thank you.
Yeah, excellent. So I have seen there are already some questions in Twitter. So let me just share with you and remind you how to ask questions to Juan. So you should be able to find a tweet that says ask questions for Dr. Juan Cubillos-Ruiz here. And please remember to include the hashtag Global Immuno, and Juan will be answering the questions via his own personal Twitter account. So thank you so much, Juan. I'm sure now you will get even more questions, and we really appreciate your efforts and sharing your beautiful work with us.
Of course. Fantastic. Okay. Thank you again.
Thank you.
And see you guys next week.
Okay, so I'll be happy to answer these questions by Twitter, okay? Thank you.
Thank you, Juan.
Bye, take care.
Ciao, ciao.
Bye, everyone.